really been a long-standing goal in ecology. I mean, people have given over entire careers to this, and in many ways quite reductionist aspects of it, but some think quite largely. And these can be anything from like gradation, competition, facilitation, etc. And to wit, the late great Bob Payne was notorious for these sort of things. Bob spent a lot of his career, in fact, I would argue probably his entire career, trying to tease apart interspecific interactions. He spent most of his time on the west coast here, northwest of us on Tatoosh Island. That's where this is. Now I had the pleasure of going out there once and seeing this stuff. It was great. Bob was actually really formative during my graduate career because he ran a historical ecology class, which he referred to as hysterical ecology. And he challenged a lot of us to challenge him. So I got a lot of opportunity to kind of butt heads with Bob, and I drew a number of black and blue bruises from that experience, but I did learn quite a bit along the way. Similarly, Steve Carpenter, pictured here looking over Tuesday Lake at the Notre Dame's research station, was also very formative on me. He was my undergraduate advisor. He gave me my first real job in this field. And the work certainly that he and colleagues have done has been very impressive on me as well. And I'll share some of that work that he's done with some other colleagues too. So I mentioned a lot of work has been done experimentally, but more recently, we've seen the ability to extract some of this information from observational data. So in particular, if you go to a location and you sample species or the environment over time repeatedly over a long period of time, you can accrue that information and basically infer some of this stuff without actually having to try to manipulate the system. This is really nice because if you were Bob Payne working on Tatoosh, it's really easy to clear off a small piece of rocky intertidal habitat, but if you're Steve Carpenter thinking about big lakes and these sort of things, it's hard to replicate lakes and do controlled experiments. So by measuring, observing things over time, you have this ability. So now I'm going to take quite a while to walk you through sort of how we go about doing this. And really the backbone of this is a Gompertz model for population growth. So I'll use this. This bar max term here basically determines the intrinsic rate of increase, So if you knock the species down very low, this is how quickly it would start to grow back again. And this term over here on the right-hand side is basically the effective density on conspecifics, or congeners for that matter. So we have basically intrinsic rates of increase balanced by this density dependent. And here's just a really simple example of what that looks like in a deterministic space. This is kind of classic logistic growth. Those systems, I argue, are not particularly interesting. You can also have some where this B coefficient here is negative and you get this 2-point oscillation. That's a very well-known phenomenon. This you rarely see in ecological studies. Very few things we can think of. The only one that I'm familiar with readily that I work on is pink sand, who have a very strong body even near southern. So in general, this B term tends to be positive, not negative. But to make this more realistic, we have to start thinking about this in the stochastic space. So the idea here is that if we multiply by a stochastic error term that we typically think represents some aspect of the environment, this drives that stochasticity that we typically observe in nature. Oftentimes, we'll subsume this term into the entire equation, and for the purposes of this presentation, a lot of other work, if we just assume those process errors, that environmental noise is Gaussian, it makes the math really nice and easy. So that's the assumption we'll work with today as well. So here's an example now of that same stochastic, or the same model where we've added some stochasticity. You can see that, yeah, there's this growth and then things kind of level around, but they're bouncing around sort of around some sort of equilibrium where that's perhaps not a point. Most conveniently, if we transform this Gompertz equation into log space, we can make this really nice and linear. So we just basically take the log of both sides of this equation. Now we have something here that looks really nice. It's just not, we removed all these exponentials. We have our intrinsic rate of increase, we have this effective density dependence, and we have our environmental needs. Furthermore, if we do just a little parlor trick here and substitute x to be equal to the log of density, now we have what's very commonly be known as an autoregressive model, in fact, a first order or a lag one autoregressive. And the benefit of this is that we have about 50 to 60 years of statistical theory upon which to rest when we're drawing inference from these types. So that's really nice. We have a really simple system here, which we can use to approximate species populations. Okay, it turns out that stochastic environment, we often have some knowledge of what that is. So typically we know something that we think to be affecting these species. So in aquatic systems, it might be something like currents or upwelling. In more terrestrial systems, in great precipitation, lots of systems, for kilotherms in particular, temperature. So we can easily accommodate those effects in this model. We just add an additional term here where our covariates could potentially be lagged so that they occur for multiple lags. And then we have the effect of those covariates, this large C. So we can still account for some unmeasured environmental aspects with WT, but now the idea is if we know some measured covariates and think those are important, we can include those. So for example, a lot of times for seasonal data, we might assume something like a sine wave. We don't actually have that measured, and you get something like this. So this is that same univariate conference model in a stochastic form where we've now forced a sine wave through there. You can see there's a sort of frame of seasonality. Okay, if we think about this model for a moment, the abundance here, log abundance X, at this point we assume is known. But the reality of the situation here is, I'm sure all of you are greatly aware, is we rarely know without error what that is. So the reality of the system is typically we're dealing with a situation where we have to go out and observe nature. Sometimes that's easy. Some of you probably seen me talk about this before in this context. If we want to talk about sockeye salmon in a stream in Alaska, it's pretty straightforward because, sorry, this is a bit washed out, but if you sit here and count these, they're bright red. They don't move much because they're sitting on reds and they're territorial, so it's pretty easy to count out these salmon. It's not always that easy though. Sometimes this is actually really quite difficult. So same picture, how many mayflies do we have? Well, this is going to mean, we're going to have to go out there with some kick nets or start turning over rocks and scraping them and do some sampling. And so clearly, to estimate the number of mayflies in just the field of view here, it's going to be fraught with errors. It's not always the counting that we get wrong. Sometimes we make mistakes on identifying. We call it juvenile coho salmon, juvenile chinook or something. We would never make that mistake, but some of us might. So the idea is that there are observational or sampling errors that we deal with all the time. And if you imagine taking a trawler or something like that out to Puget Sound and the other coast and start dragging a net around the ocean and trying to estimate, oh, I'm sure we have about this many fish, plus and minus infinity. So in order to deal with this, we can add on top of this an observation or data. And this just allows us to say that the data, the things that we've written down in our book or collected, is y sub t here, and that equals the truth, x sub t, plus or minus now some observation. Okay, together, these form what's called a state space. Again, we have a long history of statistical theory upon which to base inference for state space models. But it's really nice now because we have a relatively simple model for population dynamics, and we have a model that allows us to deal with this kind of observation set. So here's an example here where the true state of nature would be the gray trajectory, and the observations, the things we write down in the book or record, are the blue. And if we make a long enough time series, we're able to actually separate that observational error from that processing. And if we have replicate samples, then we can even do a much better job. So this is a warning to all of you, or a note of caution if you're considering doing these sort of things. I would strongly suggest 2 or more sites at every location because it will create you a lot of additional power trying to infer these interactions. Okay, that's population level. I started out by talking we were at least thinking about 2 different species and oftentimes many more. So now we want to start thinking about community dynamics here. So if we think about a community of animals out in some food web, about aquatic food webs in a moment, we typically think of the number today as being a function of the number yesterday. So how many births and deaths give us basically an immigration, there we are. So we want to account for the number yesterday. We want to account for things like predators, things that eat animals, the things they have to eat, their prey, and their competitors as well. They're out there competing for space, mates, these sort of things. Then, as I mentioned earlier, we've got a lot of these external forces, basically the environment that's driving these systems, pushing them around, and sometimes those forces can be really strong. We're going to see later on that they actually are really strong relative to other. So to account for those, we can modify this Gompertz model by now, instead of treating it just as the I you might think of, we have a sum of all the other members in the food web. So here we have n different members, and the abundance, log abundance of some taxa I is its intrinsic growth rate of increase plus or minus all those other effects of the animals in there, plus some environmental noise. And when I equals j, this is the effective density dependence. So now we can estimate the strength of density dependence as well as the things like effective predators, prey competitors. Okay, that model we can write in multivariate form more compactly as a matrix and vectors. So here now we've got a vector R that contains all of the species-specific intrinsic growth rates, and we have this interaction matrix V, and then we have a vector of environmental process errors, which typically we will assume are multivariate normal. Again, this makes the math really nice. We don't have to do that, but it's convenient. And then there's a covariance matrix Q. So this model now is what we would call a multivariate autoregressive lag. Okay, that covariance term, we can assume really simplistic forms. So in these two examples, there's no covariance. The example on the left would be what we would typically think of as independently and identically distributed. So they have the same variance for all species. The one on the right would be independent, but not identically distributed. If we think there's a lot more going on there, some interactions among those species that we can't account for with our covariates or our error term, then we can include covariance effects as well. So we might have a case on the left where it's if they're identically distributed, but they all depend on one another, or a case on the right where everything's kind of removed. We also can set up much more custom forms of these matrices. So if we know we have different species in there from the same genus or different functional groups that we think the environment are going to be pushing around more or less similarly, we can create any sort of form of covariance because we like. Still has to be positive definite and violate sort of fundamental rules of math and statistics, but putting on that ecology hat and really think about what's going on, you can create much more realistic representations. So again, we can include the effective covariates here. So things like temperature, currents, precipitation. So now we have an M by P matrix C, which are the effects of those covariates. And then we have a vector, small c here, that contains those covariates at different lags. And H here I've written kind of cheated a little bit. Not all those lags need to be the same for each covariate. You know, something that was 1 month, something that was a year, something 16 months.
Do you have a good practice for identifying what the appropriate lags of those are? Is there best practices?
Well, best practices. Oh, sorry, for those online, there's a question about if I can recommend a best practice for selecting appropriate lags for covariates. If you want to sort of cheat and do some data snooping, you could just do a quick cross correlation between those things. Here, we're not doing so much hypothesis testing. We really are trying to do it, so it doesn't give me so much heartburn as it would someone else. The other thing would be to rely heavily on what previous literature or expert advice would tell you. Yeah, I see this, and then 2 months later, I see something else, or something about generation times, that sort of thing. The other alternative is to fit a lag, estimate something like AIC at a different lag, so do it that way. So set it up. And you got to be careful because at some point you'll find something that's the best, and this is an issue we address often. But if you're looking at things like a cross correlation function or the like, 1 in 20 of those are going to be significant by chance. So oftentimes people get, oh, I got this crazy thing going on at 8 months. I don't know, what does that mean? I don't know, it's significant. Well, it's probably just spurious in that case, so you gotta be a little careful about this. So again, put that ecologist hat on and think strongly about it. That's a great question and something that people wrestle with all the time.
I have another
Yes,
question, which is, can you explain to me the ecological assumptions of that B matrix being exogenous, like fixed parameters?
Oh, sorry. So here, the elements of B being fixed as opposed to like time varying
Or being a function of, like, I guess, relative density or of the different species.
Yes. So question was, what is the rationale for assuming all of these elements of this interaction matrix are fixed or unknown or potentially random, but not functions themselves? And in this case, it's purely a mathematical. The reality of the situation is these probably vary over time as well, and probably vary as a function of things like density and whatnot. So yeah, so this group that I've been working on, we have some models where we've actually got a T subscript on there, and then you treat the B matrix itself as some sort of autoregressive process. So maybe on average, there's some predation rate, but it may be higher or lower depending on some other aspect of the environment. So in this case, it's really just a convenience. So yeah, it's one of the many assumptions we have to make. Assume some sort of semblance of reality. Okay, so then importantly as well, the effects of the covariates on those different species could be different. You might subscript those as they are here on the left case, or they could be the same. So if you have a lot of different species with very different biology, you might assume these things are different. But if you had many organisms that were very similar biology, you might make the assumption that they're the same. And these two options are typically something we would do similar to Cody's question, fit different forms that our own has for the data support. So here, for example, right, was a case where I got two covariates, temperature and dissolved oxygen. They have some lag. So you may fit different forms of capital C here and then try different sort of value, which you think is. But again, we want to start based on first ones. Okay. Just as we did in a univariate case, we can form a state-space model here. So now the idea is that we have a vector Y, which are the observed data. And those are going to equal the true underlying states, the X, plus some offsets. The offsets could represent something like varying survey effort. In one place, you surveyed a fourth of an island, another spot was a half of an island, or something like that. And again, here we'll assume that those errors, the observation errors, are going to be multivariate. Similar to the case with the process errors, we can assume different forms of observation errors. But if you're not careful, you're going to quickly end up in a place where it's really not identifiable. So typically, we try to make that process covariance matrix as rich as we can, and then assume the observation errors are not correlated. Now that said, if you had information from previous studies or secondary studies that showed that if you make a mistake counting one species, you're pretty sure to make a mistake counting the other one. Especially if there are only two options, if you overcount one, you're going to have to. So if you have information about that, you could put that in. You don't always have to estimate this. Right? In a lot of cases, people know from other studies, they have a pretty good idea of what observation variance is. They've done repeated measures or just have different people come and survey the same particles, habitat, that sort of thing. Oh, this would be one that you actually could probably get because there's really only two terms here. You have one covariance term and one variance term. This pretty much you're not going to get that unconstrained, especially if you have an unconstrained process covariance matrix. Okay, so now, given we have a way of estimating different species interactions, I want to talk about some different metrics that we can use to estimate the stability of these communities. So we're thinking now, what is it about the way that these individuals are interacting with one another that either makes a system stable or unstable or or what makes it relatively stable compared to the. So we're going to walk through some of these. And the three I'm going to talk about today are the rate of return to equilibrium, something called reactivity, and then the other one has to do with the variance of stationary distributions, these interactions. This will hopefully make a lot more sense in a little bit. So first of all, I want to talk about rate of return to equilibrium. So thinking way back to the beginning of the talk, we were talking about deterministic models. In deterministic systems, we generally have two forms. One is a point. So in this on the left, it's that X star, Y star point. So if you pull the system away from that, it will come back to it in some manner. The other is sort of a stable limit cycle. So there's, you pull things away and then they'll run around in some sort of phase. And we have a long history of studies that focus from both a theoretical as well as an empirical aspect to this. Now, that said, I sort of motivated this whole thing by saying these models are largely garbage because they don't really represent reality at all, much to the extent that any model can. So when we're thinking about stochastic systems, we're really thinking instead about the equilibrium being some sort of distribution. We call it the stationary distribution. So we're thinking about systems that will achieve some kind of asymptote, but they're going to oscillate around in a random fashion. So if we go back to our univariate model, if we're willing to make the assumption that that density-dependent term is less than 1 in absolute value, this AR1 model is what we call stationary. That means it's essentially mean reverting. The variance will not change. The mean does not change unless you force it so from the outside. And if you would think about the long-term properties of this and imagine X at some time way into the future, think about it as infinitely far into the future, you can construct a distributional form for that stationary variance. So here, you at infinity, t and t minus 1 are equal. So that's where we get this aspect. And I'll show you later the proof of this. But basically, the mean over the long term is going to be this intrinsic rate of growth divided by 1 minus v. And the variance is going to be that process variance, the variance from our error terms amplified by those species interactions. I'll show you a schematic of that. So here we have two different examples. The one on the left is where the density-dependent term b is relatively small, it's a half. And that actually represents fairly strong density dependence. It's a little bit counterintuitive. Your smaller numbers mean stronger density dependence. The one on the right is 0.9, so much larger. What I've plotted are 10 different trajectories from the exact same model, all starting at -7, both sides, left or right. And what you can see is the panel on the left. They start down here in the lower thing, they jump up, and pretty quickly they're oscillating around 0. Panel on the right, you can see that some have gotten there pretty quickly, but others, like this one down here, takes a long time to get there. So in both of these cases, the distribution of the process errors, the variance on Q, is given by that gray density thing on the margin, and the stationary distribution is given by that color. So in this case, the stationary distribution is nearly the same as the process variance. Over here, though, it's much broader. So the idea is that when you have a relatively weak independence, these populations wander all over the place in space, and they're not very stable, so to speak. Okay, the transition distribution is an aspect of this. So the transition distribution we think of as at some point in time, not the sort of long event horizon. And basically, we can construct a form for the mean. So the mean here says it's basically the long-term stationary mean plus this effect of that density dependence, where this is raised to an exponent. And you're subtracting a starting point, x0 minus this. So you can see here, b is really small. This term is going to go to 0 pretty quickly. It's going to get fast. The variance here is going to be that stationary variance modified by 1 minus b squared raised to an exponent t. And this is perhaps a little bit more intuitive if you think about that rate of convergence being governed by. So here is the plot of that. So again, case on the left is what I showed you 0.4, B is 1/2. Side of the right, B is 0.9. And you see that the variance and the mean of the transition distribution get to their long-term, the stationary mean and variance very quickly when it's 0.5, and when it's 0.9, it takes them a little longer. So you can think about the transition distribution is where we are now, and the stationary distribution is where we're headed. Okay, so when we're thinking about these multivariate community models, then that rate of return is going to be governed by that interaction matrix B. So it's the effective density dependence, and then all of those predator-prey competition interactions. And just as it would if we were doing this with a deterministic system, it really depends on the eigenvalue. So it turns out that the rate of return for the mean is going to be given by the maximum eigenvalue of that matrix B. So here lambda sub B is the eigenvalue vector. You take the maximum of those, you'll get the rate of return to the mean. For the variance, we need to use a Kronecker product. A lot of people are familiar with Kronecker products, but basically you could think of it as sort of the outer product for vectors expanded to matrices. So if you have two matrices, A and B, the Kronecker product says you take all the elements of A and multiply that by B. So you end up with the dimensions of this times the dimensions of this. So the rate of return for the stationary variance is given by the maximum eigenvalue of the Kronecker product, B. So you have to multiply these two together. So you can think of this, in the univariate case, it was B squared. We can't square a matrices, so this is essentially a square number. Okay. Rate of return is really based, this notion of this is really based on sort of long-term properties of a system, this idea that you have this infinite time horizon and you can perturb the system and wait around forever for it to get back there. In many applications, probably most of what everybody in this room and online works on, that's not very reasonable because before this, after a system is perturbed, it starts returning to where it is and then it gets hit again. And it gets hit again. Gets hit again. So rarely do they get an opportunity to get out to that sort of long-term horizon. So that motivates this notion of what's called reactivity. And reactivity you can think of as the displacement of the community away from some sort of, in this case, a stationary distribution. I'll show you an example in 2 space. I can't envision it in higher dimensions. So it's kind of, you can think of it as sort of a balloon. When you disturb the system, you blow into the balloon. If the balloon is really stiff, you can hardly push that thing out, but if it's relatively weak, you blow the thing up, right? Just one breath. So here's an example of reactivity. On the left is the bivariate plot, species 1 compared to species 2 in both cases. On the left, it's is a less reactive system. On the right is more reactive. So the idea is after a disturbance, the system on the right is moved further away from sort of the origin, 0 point. And its return back is a little bit shorter at a subsequent time step. So the arrow, the base of the arrow is time t, the tip of the arrow is time t plus 1. So this is the idea of reactivity, how this is sort of blow up in multivariate. So let's now go back to our model. We're going to ignore covariance for a little while. The math turns out it's the same. And imagine now we're going to give a small displacement, we're going to call that vector delta, to our vector of abundances, x. So you just do the math and you can see here, now that the magnitude of that response is given by b times delta. If you want to know what the response of that is relative to the displacement itself, so if we really hit this system hard, based on that shock to the system, how far does the system itself move? It's just the ratio of those two. So the length of those, that matrix product is a square root. Again, you can't square these things, so it's a transpose times the matrix itself, divided by that. The reactivity then is given by the largest singular value of this matrix.
So, quick question.
Oh, yes.
So is your definition of reactivity different from the concept of resistance in dynamical systems theory?
Yes, well, that's a really long answer. Whoever that is, I'm a Bible years. The literature is ripe with semantics problems, and so as you follow along, people will switch terminology somewhere in there, but it is in some ways analogous. It's not a problem, is that a lot of people are using resilience to refer to resistance. This is where at some point that was again a time switch that someone decided they would start doing that. It's really confusing to me. Okay, so this sigma max here, I apologize, it's not a variance term, it's the largest singular value. You take the log of that, that's reactivity. If the reactivity is greater than 0, the system is said to be reactive. And that means that when you perturb it, it has the possibility that it will actually amplify that perturbation. If it's less than 0, it will be hit and very quickly come back. And you don't typically see some expansion of the system overall in that multivariate space. It turns out that, maybe this will help answer the question, there's a bunch of mathematical equivalencies here. So the definition that Newbert et al. Came up with was largest singular value. It turns out that that's also the L2 norm, which is also the square root of the maximum eigenvalue of the transpose. So if you're looking around the literature and you start to find these different answers, that's why. Okay, so last aspect I'm going to talk about here is the variance of the stationary distribution relative to process variance. So in a relatively stable system, the species interactions are really only going to amplify that background variance a little bit. And you saw that with the univariate case where B was 0.5, the two distributions were really quite similar to one another. But in a less stable system, all these species interacting with one another will tend to amplify that. And or if there's weak density dependence. So all of those things will interact to sort of make this system behave, it gets really squirrely in a hurry. So let's go back to our univariate case because it's a little easier to think about. So here we've got one species, we've got our stochastic confidence model with Gaussian errors. That stationary variance is here, and I didn't define it earlier, but you can do the math and come up with the fact that it's the process variance Q divided by 1 minus B squared. So again, we're assuming that B has to be less than 1 in absolute value. So B is relatively large. This is pretty small. That gets big, which you saw in the example earlier with the contraction. So in a multivariate case, we basically go through the same type of math, but we arrive at a problem pretty quickly. And that is this term for the stationary covariance, sigma sub infinity, is not closed. There's no way to solve this equation for sigma. So we reach into our tool bag of tricks here and we pull out something called the Vec operator. And all the Vec operator does is turns a matrix into a long column. So if you're willing to invoke the Vec operator and define an n by n identity matrix I, then the Vec of that covariance matrix, so decompose a matrix into a long vector, is going to be this Kronecker product I minus the Kronecker product B, that inverse times the covariance. So now we're thinking about all of this stuff going on in B here driving this. It's analogous to the univariate case. So if all those values in B are relatively small, then it's going to be relatively well behaved. If they're all relatively large, it's going to be large. Okay, so we found a term now for our variance term, either in univariate or multivariate space. Now let's go back to the univariate space. So here we have this term for the stationary variance. It's Q over 1 minus B squared. If we want to think about the proportion, we'll call this pi, of that total variance, sigma infinity, that's resulting from the species interaction with itself, strength and density dependence. We just do a bunch of algebra, and we see ultimately that here's the stationary variance minus the process variance divided by stationary variance. This is a proportion owing to density dependence. It's just simply B squared. So in the univariate case, When B is relatively large, the proportion of the variance owing to, or the total variance owing to the species interacting with itself is relatively large. That was that B 0.9 case I showed you. Okay, the multivariate case is analogous, but we have to invoke a little more math here because that scalar B, the size of that is just itself, right? What is the size of 1? It's 1. But in a multivariate sense, we need to figure out a way to sort of measure the size of a matrix, not the dimensions, but what it really means in sort of, high dimensional space. 2 space or 3 space is relatively easy, but higher dimensions is hard. So to do that, we're going to talk about matrix determinants. So this is the really easy part of today's talk. What is the volume of a cube, each side of which is 2 units? It's 8 units, right? 2 times 2 times 2. Okay, we can instead define that cube using 3 vectors. Set up one vertex, it doesn't matter which one, you pick that as the basis. And then each one of those vectors describes the side of the cube. If we put each one of those vectors into a matrix and ask for the determinant of this matrix, it will give us the volume of that cube. So for those of you who don't know matrix determinants, works like this. If you have a 2 by 2 matrix, the determinant of this is going to be AD minus BC. And then if it's 3 by 3, you have to do this multiple times. So you have A times the determinant of this, minus B times the determinant of that, plus C, etc. So you go through this math. So you get there. If you want to go through all the long math to prove that this works to yourself, you can do that. If you have an R console there, you can just ask. Also, I should point out that the column ordering here doesn't matter. I chose this so they were all on the diagonal, and you rearrange that so they weren't the determinant of that matrix is still 8. Okay, so now we'll use determinants to tell us sort of the volume of this matrix. How big is this sort of space that it's on? So we have our term for stationary variance. And here we're not going to worry about solving this for sigma, because we can make the math work without it. So now we want to know the difference between sigma, that's the stationary variance, and Q, which is the process variance. What's left over is going to be going to species interactions. Okay, so if we take the determinant of both sides and do the algebra, you'll see that the proportion of variance owing to those species interactions is going to be equal to the determinant of B squared. So in the univariate case, it was B squared. Here is the determinant squared. Okay, so one question I usually get about this time is, okay, a lot of this is really just talking about the community as a whole. This is stable, it's not stable, it's more stable than another, that sort of thing. But typically, I personally often want to know, well, who in this community is leading to that most, greater stability or instability? So oftentimes we're thinking about, okay, what's the particular interaction we're interested in? Are intra or interspecific interactions more important? Are things like top-down or bottom-up? Right? So you have this matrix B, and it tells us all those different things. You put all your players in there, strength of density dependence, competition, predation, that sort of thing. It turns out that you can basically say, well, we know what that proportion is, that's the determinant of B squared, and we want to know per unit change in an element of that matrix, B sub ij, how does pi here change that proportion? And if you do that derivative, you get this term. Those of you who are familiar with this, if you took away this square term right here, that determinant, this right here is Jacobi's formula. Cross that. So anyway, now we can figure out, okay, which of those interactions in this matrix is really leading to the biggest change in the stability. Okay. Finally, we're going to get to some results. Okay, so how do we actually do this? For those of you who use R, there are several packages out there that will estimate things like capital B, that interaction matrix, capital C. Many of you are familiar with these. One package in particular I'm fond of for good reason. You also have the ability to code these sort of problems yourself. So if you were good at this sort of thing, like JAGs or STAN or TMP, and you just code it yourself, it's not really difficult. The advantage of using one of the code your own languages is you don't have to assume Gaussian errors, especially with that data. It's like a negative binomial or something like that. You've got count-based things, and we actually have a package that works on STAN to fit these kind of models. Okay, so now we're going to get to some actual ecology, and I'm going to start with the Lake Washington story. And this story starts with the late great Tommy Edmondson, who was a faculty member in the Department of Zoology for many, many years. It's not even the Department of Zoology anymore, it's biology. And back in the 1950s, Tommy started here at University of Washington, and right outside his window, more or less, there were problems in Lake Washington. And if you pull headlines from the news, you know, lake water's in bad shape, lakes stink out to clean up. And the problem was that the city of Seattle was dumping raw sewage into the lake. So this was the era of dilution is the solution to pollution, right? So raw sewage has a lot of nitrogen and phosphorus in it. Nitrogen and phosphorus is a great fertilizer. So what was happening is you would get massive algal blooms, so much so that it would turn this system from a typically freshwater or a phosphorus limited system to a nitrogen limited system. That favors blue-green algae, cyanobacteria, because they fix nitrogen. So you have these just nasty mats of green filamentous algae fluoride. Stuff was dying, it literally stank. So Tommy, through a bunch of his work, convinced the city of Seattle and the county to stop pouring sewage into the lake, and they diverted it now to sewage treatment plants, and the lake has really gotten much better over time. So here is a time series of, on the top left there, Secchi depth, the dots, and Secchi depth is an index for water clarity. So it's basically, for those of you who don't know, it was once upon a time a dinner plate devised by an Italian named Secchi. He lowered it over the side of the ship. Doing this. Nowadays they paint, well, it's kind of the four quadrant. You can see this over here. 2 white, 2 black. You lower it down until you can't see it anymore. You pull it back up till you can't see it. The average is 2 depths, and that's what it is. So early in the 1950s and 60s, Secchi depth was about 2 meters. More recently, it's sort of like 6, 7. So the lake got a lot cleaner. Concurrent with those changes, so here's the sewage time, here's the diversion, and now there's no sewage going into the lake. Here's that oscillatoria, is one of the dominant blue-green algae. So this species was off the charts. You would probably all look long across the lake. There was a large invertebrate predator, neomyces, that was quite dominant in the lake. Then longfin smelt invaded. Neomyces were away because longfin smelt ate them all. Now we've got a whole history of other invasions going on in the lake. It's really kind of a hot mess. But as an ecologist, it's pretty fun because you've got all these natural experiments. And then there was rise of Daphnia, which is a really dominant race. Many people refer to Daphnia as the Holsteins of aquatic environments. Really almost a Canada geese as well. So based on all of Tommy Edmondson's work in his lab, Stephanie Hampton, who was a postdoc here with Daniel Schindler, and I worked on a paper, and we came up with this diagram on the right. This was based, again, on a lot of observational experiments, lab experiments, mesocosm experiments, etc. And this was sort of conventional wisdom. We had Daphnia really driving a lot of what was going on. So in this case, black solid lines are top-down effects, dashed lines are bottom-up. So phosphorus was feeding into oscillatoria, it was having negative effects on all these things. So we got our hands on all this data, finished some of these models we were talking about, and looked at some of the different players out there. In that paper, we didn't address stability. We really focused heavily on the species interaction. That was where Stephanie's heart lied at the time. So now I'm going to go back and revisit those data and compare them with a different system. So for this analysis, I'm considering 2 different time periods, both of which are 6 years. The first is 77 through 82. That's right after the sewage diversion stopped and Daphnia had risen. And then the last 6 year period here is 89 through 94. That's the data that I have readily available. Stop that we could update this analysis much more contemporarily. also, you'll see in a moment, compared these results to a long-term study that Steve Carpenter and colleagues have been doing in Upper Michigan. I worked on those lakes. I counted a lot of those plankton actually as my first job out of undergraduate. So I'm quite familiar with that system. It also represents a quite undisturbed area. It's inside a large private research reserve. So there's no urbanization. There's really no land cover effects, no harvesting of trees, no agriculture, that sort of thing. So in order to make this analysis comparable with the other system, I have 4 different functional groups here. So there's small phytoplankton, which are generally about 30, 35 microns or less. Large phytoplankton, Daphnia, this whole scheme of aquatic systems, and then all non-Daphnia. So we've got 2 food sources, 2 grazers. They're really to compete with one. Here are the data. On the top is large phytoplankton. That's log biomass, so those numbers are huge. Small phytoplankton is the next, Daphnia, and then non-Daphnia. And here I'm showing you the entire time series, just so you can see an idea of what's going on. But you can see, first of all, a few things pop out to me. Largely no trend over time. We really are stable, so to speak. But you see really strong seasonal patterns. So every year there's a sort of rise and fall of phytoplankton followed by a rise and fall. So I mean, it's not always the same time, but there is a really strong seasonal effect. This is really well known for pretty much every north temperate lake because we have strong seasons at these latitudes. The weather affects this as well. So temperature is a large driver, but also phosphorus. Most of our phosphorus centers here do runoff. So in late winter or late fall, winters when we get all our reeds, phosphorus is arriving. The covariates I'm using here are water temperature on the top and the amount of phosphorus in milligrams per liter. Little bit of change in phosphorus over time, but again, you see what's really dominating here, these really strong seasons. So the hope is that these covariates are going to pull out that seasonal signal. They'll also have some effect on phytoplankton. I don't allow phosphorus to affect the zooplankters because they're grazers, so they no fertilizer effect. Okay, so to cut to the chase, here are the 4 stability metrics I discussed for the early period, the late period, and then the Ives et al. Analysis. This is from that North Temperate Lake, Michigan. What you see is that the late period in Lake Washington is more stable than the early period. This number is larger, so these smaller numbers here mean more stable. Both periods in Lake Washington are what we would call reactive, whereas the system in Upper Michigan is not. So anytime we hit this system with something, that amplitude grows initially before falling down. And this Midwestern lake is sort of intermediate in stability to the early and late periods. Notably here too, if I had asked everyone ahead of time, the percent of the total variation in the system, that stationary variance owing to species interaction, is like 1 to 4%. When I first started doing this, I was sure I had made a mistake. There is no way it's that. And it turns out that Machinda, Bob O'Hare, and some other people did a meta-analysis published about 15 years ago now, and found that across a whole bunch of ecosystems, vertebrates, invertebrates, you name it, they found that species interactions accounted for about 5 to 60%, but in fish, in an aquatic system, it's 2 to 10%. So what this says is those things can be out there fighting each other, eating each other, doing it. It doesn't really matter. They're on a roller coaster that's zipping up and down in the environment. They're being dragged all over the place. So that's often the way I describe these systems, right? It's kids on a roller coaster looking at each other, then they're flying up and down. Sort of their displacement doesn't matter. It's sort of what they're doing. It's all environment. Okay. Then I looked at the sensitivity. It's a little hard to kind of visualize these. You have to look at a whole bunch of matrices and stuff. So I'm just summarizing the text here. That early period in Lake Washington, most of that change was really sensitive to bottom-up effects of phytoplankton. More recently, those sort of effects tend to die down. They're all really modest and not very interesting. Really close to zero and kind of spread across the board. In those Midwestern lakes, they found that it was really density-dependent effects that have the most influence on what was going on. So it was basically in animals competing with one another. So in summary, hope I helped you see that a lot of times we focus food web studies on species interaction, about who's eating who, who's competing with whom, that sort of thing. But in the reality, especially in, I think, a larger management context, knowing something about the stability of the systems is important as well. And we have a number of tools available. With that, I can keep.
Yeah, does the temporal resolution of your data alter your inference from this sort of model? Like it looks like, yeah, lump things together in month, week, year, however. Does your inference change, or do you have a preference on how you approach that?
Yes, it will change, first of all. So these were all monthly data that I was using here. If you folded those up into annual data, all those interactions are going to vary differently because now you're integrating over a long period of time. That's also a really important point because some of these systems, the temptation is to build a model that has orcas, pinnipeds, salmon down to like diatoms, and the generation time of a diatom compared to an orca. Right, so if you're thinking about how much of these, what do you really get in a week or a month? I mean, from an orca, right? It could be a couple generations of fighting. So the time, the resolution of the time is important for, number one, just to estimate those interactions, but also if you're thinking about the system as a whole, you really have to sort of think about it, ecological perspective. Some people who are a lot smarter than me have worked on these models where they use a continuous time, so you put all the differential space or a discrete space, and those you can start to capture some more of that variance in generation times. But I feel like that is still an area that's really ripe for more research, sort of explore what are those effects, assuming different time. That's really a question. And Cody, I think you had a question?
Two. The first one is a goofy one. If fish are kids on a roller coaster of environmental variability, do you think that makes fisheries managers' parents screaming at them ineffectively from 3 rows back? How does that, it's kind of a goofy question, but it's also a serious question, because if that is the case, what are all of these fisheries managers doing?
Well, I mean, I don't do the type of work. So I'm probably not with it. But my understanding is, right, that was a huge advance in stock assessments when they actually started giving explicit consideration to the environment. Right? I mean, they were like, hey, this time we're doing cold-blooded animals. You got to consider temperature. Yeah. So I think there's that is often now explicit, if not implicit, in what's going on, that people have a really understanding about the environment and how biology interacts with the environment. And they know when animals are spawning or breeding relative to what's going on in the universe, that sort of thing. So having some idea about what's going on there allows you to sort of frame that within appropriate context so that you're thinking about that in the background, if not explicitly, implicitly. But I think, we saw some stuff yesterday. I think fisheries management still has a huge role in what's been done, because these systems that I'm talking about here, we haven't done any of that harvesting aspect as well. So, right, I mean, yeah, imagine if we went back to kind of pre-colonial times or something. Infinite knowledge of what was going on out there, it would look very different than it looks now. But that's a really good question. I don't have a great answer.
That's fair. Second question, if you don't mind. So one of the reasons I'm here today is because I'm trying to do a very similar thing with species in the Bering Sea. But it seems like one of the key, one of the focal points of many of your analyses is stationarity, and the thing that we don't have in Bering Sea is stationarity. And I'm wondering if you have any suggestions or thoughts on directions to go when looking for species interaction, community interactions in non-stationary systems.
Yeah. Well, right. I mean, so that we see that a lot. And if you that in a univariate case here, let's just not even worry about community interactions. If that B term, as that approaches 1, that's a random walk. That's not at all stationary, right? So once you go fall over that precipice, all bets are off. Everything looks different. So is the non-stationary aspect that is it more like trends, or is it about step changes and things like that?
Both. It depends on what species you're looking at. one of the things I've been playing with is just doing hierarchical clustering of a time series to see what kind of wiggles together. And there are some that just kind of trend up over time, some that trend down over time, and some that do stair step things. So
Yeah. So what we typically try to do there is those time series I showed you were not on station. That's seasonal signal and that is not station. So that's why we added those covariates, temperature and phosphorus, which had a very similar pattern. So once you look at the residuals of that model, it does look like that. So you pull all that stuff out. So, you know, in your situation, you might be wrestling with, you know, some aspects, some different waves of things, and that's why we often will use a discrete Fourier sequence, right? You just set some with different phases and wavelengths, and you can try to tease that apart. Add some linear trends, add a quadratic trend, so just add a bunch of those terms in there. So that once you pull all that stuff out, what's left looks to be relatively stationary, and then deal with it there. Alternatively, you start moving towards something that I alluded to with Sunny's question. Where you start thinking about like that interaction matrix is changing over time as well. Then you start thinking about, okay, how can we do that? So the question, you know, some people have done that with moving windows. Take a window that's small enough that you can still estimate all this stuff, and then just march that window along. They're not independent of one another, those things, but you still can often see what's going on. And that's what's being done a lot in the ecological literature, where people are looking at early warnings of regime shifts. That autocorrelation coefficient in the univariate case, they just do it over time, so you'll see the thing starts low and it starts to creep up to a random walk, and pretty soon, yeah, it's just running all over the place. Or it will undergo some catastrophic change. So happy to chat about this.
yeah, I'll come bug you again at some point. If you don't mind.
We have a question online from Eli Holmes. Eli says, for the plot regarding species interactions accounting for 2% of reactivity, was that for reactivity or for variability?
It's,
Meaning, was the point that 2% of the variability came from species interactions?
no, it's a change in a proportion. So it causes a 2% change in the proportion. So you're going from, you know, 1 to 1.02 sort of thing. Yeah.
That's all the questions on that.
Okay, great. People should leave. I know I kind of.
Yeah, it was, what other factors would be confounding those beta coefficients? Because my impression is that they're the correlations between the different species abundances at a certain point in time. But would there be other bits confounding that?
Okay, so just to make sure I understand, that the elements within that B matrix are to some extent estimating the correlation between those things. Yes, that is a big part. That's essentially how we're estimating those, right? Is yeah, you add that up across lots of different things and try to figure out who's going who. And then remind me a second for the actual question. No, I'm sure.
Would those beta coefficients, which are correlations, then be confounded by other factors that are not species interactions?
Like you mean covariate effects, that sort of thing?
Yeah, or like they have like R-beam, or there's like some, like, say, temperature impacts one species one way and another species one way, but because you don't have temperature in there, you're then sucking up that you're assuming then that is correlation between the two species rather than some third force.
Yes, yeah, I mean, absolutely. It's still essentially a linear regression problem that its root response is not independent of the independent. But yeah, so if you're not accounting for, right, it's just variance partitioning. So if you pull something out of one term, because the variance on the left side of the equation isn't changing out. So yeah, it's how are you partitioning that variance in the box? So that, again, is a reason why it's really important to sort of think about what you're using to explain the rest of it. So I didn't show any of this here, but we spent a lot of time looking at residual plots. Yeah. Some structure left over in residuals when you're done, so do that. some people have even thought about, well, let's not even worry about the species interactions first. Just fit a bunch of the covariate stuff to Cody's question, so you don't see those really strong seasonal patterns in there. Get that out, then feed what's left in there to try to estimate that. I mean, the other people have paired these up with some occasions where they actually have pretty good experimental data to see what's going. So if you have a long-term study where you have some idea what's going on, but things you can actually observe what's going on, you can take the thing up, puke it, that needs something else, try to stomach something, that sort of thing. Yeah, then you'd have yet another piece of sort of truth that you could evaluate against. But yes, it's not a magic, sort of silver bullet here that's going to get you around all those other things you have to think about at the time.
I have a conceptual kind of question. So you talked about in the covariance matrices about, like, okay, got to worry about not having identifiable issues and stuff like that. I wonder, is that because, so you have like some matrices where you have on the diagonals were the sigmas, and then the off diagonals were like gamma, like 1, 2, and then gamma 1, 2. Would there be a way to have like gamma 2, 1? Is that what you were talking about with the identifiability, or because sometimes there's not data to explain those interactions?
Yeah, well, and then it's got to be symmetric.
Okay.
That matrix
Yeah.
has to go 2, 1 has to equal 1, 2.
Yeah.
But yes, it's just that because in that state-based framework, right, you have a signal, and you think that that's resulting from some true process and then some additional variance. So if you're trying to separate which of that is process variance and which is observation variance, you need a long time series to potentially replicate. But even when you're doing that in a multivariate context, you're not going to get all of that. It's just too hard to pull all that information. Maybe if you had like unbelievably long time series. The other thing, the real root evil here is that it works best when the process variance is much larger than the observation variance. It's rarely the case in ecology. Rarely the case. It's almost always that the observation variance is much larger. And there have been papers that have shown how Kevin C. might work on some of that.
Any last other questions?
Did you have something? No, oh, okay.
Well, I guess with that, thanks everyone for staying online, and thanks everyone here. So, and thanks, Mark.